Okay, today I'm going to talk about a project we started, my lab started during the lockdown with Zoom. And this was work by Van Betauer, a master student in my lab. This was one of 4 of his projects, in fact. So I don't need to explain to this community why Candida albicans is bad. Part of its pathogenicity comes from the fact that it can shapeshift, that it takes on these different morphologies. So they can range from your classic yeast vegetative forms to filamentous forms like hyphae or pseudophyphae. And basically these different morphologies confer different cytoprotective properties, and that's why partly why it becomes a life-threatening disorder in some humans. Okay, so in fact, there's something like 12 different catalog morphologies of Candida, and today's talk is just going to focus on 9 of these morphologies, so they're somehow more ubiquitous, or at least the ones that were a bit easier to grow in the lab. Okay, and if you don't take any other thing home from this talk today, the main message is that we have software called Candescence that solves the following problem. It locates each object in this image, okay, and it then predicts for each such object what it believes to be the morphology. And looks something like this, and you can see that this cell here is labeled as yeast white, and we even get a confidence score there that's 0.82, which means it's quite confident that this is a yeast white cell. And of course we hope there's not too many examples like this, a false negative, where it's missed this cell here, and we hope there's not too many examples of this case here where for some reason it's labeled this as a germ tube. That's a bit strange, I'm not sure why. But the bottom line is that I would give it about a grade A- in terms of how well it performs, but this was the first shot at a very complicated problem. And you can download the software and all of the data that went into creating it, all the code, everything else. It should be entirely reproducible via the Open Science Foundation website here. And our bioarchive manuscript should be available sometime next week, I believe. Now, if you're going to stick around for the rest of the 10 minutes of the talk, I wanted to basically provide an overview of the deep learning paradigm, and so that perhaps as I'm speaking you could think of how you could use deep learning in the context of your own research, and we're happy to collaborate and to help you bring deep learning as a more day-to-day entity into your research ecosystems. So it usually starts with a compendium of examples that are curated, gold standard in a sense, right? Your learning set. And towards that end, we built a compendium of C. albicans DIC images. In fact, we grouped we grouped lots of different colonies and took about 1,200 images across different strains and conditions that we knew induced these 9 different morphologies. And then via Zoom, we started to label individual cells, about 9,000 in total, across these 1,200 images. So you can see here that we've labeled some cells white, the red, some cells yellow, and we labeled some cells this green, gray-like. So that's not actually gray cells. We can't say that. We don't have molecular markers, and that is how ultimately it's defined. But we did notice that there was a significant number of cells that looked neither classic white nor classic opaque, and so we decided to label them gray-like. And we labeled SHMU and also things like technical artifacts from the microscopy itself. Filamentous cells were a bit more difficult, and so we helped Candescence learn a little bit. So by labeling not only the entire filamentous structure, but also the start and the junctions, because the junctions are visibly different between hyphae and pseudohyphae. And so we hope that these kinds of extra clues would help the system out. Okay, so this is kind of what a typical marked up file would look like. You can see all the cells have been labeled with their putative morphology. And it gets more complicated. These markups look a bit more complicated. Here we have some hyphae, and you can start to see a lot of overlap. So that's basically this data set called Veracina, right? And is what we use in the training of the deep learner. And okay, I would highly recommend that you go to YouTube, and there's so many nice videos these days designed at for every different technical level, whatever your background is, to understand some of these concepts from deep learning. Let's suffice it to say here is that we have this kind of strange network structure, and we feed our images one by one into this network structure, and they percolate down through the network. At the bottom, we predict where and what type of cell we see. And that information of successes and failures to predict correctly is back propagated back through this network and updated. And what we repeat this process of passing examples with known answers over and over again in what are termed epochs, until we hope that this just converges on a good classifier with nice performance. In fact, we chose something quite modern called an FCOS, and I won't go into the detail, that solves this two-step problem of both detection and classification. And we used our Verostina data set, and then we did something kind of nice there where we stratified our data set by elementary grades, so grades 1 through 6. In grade 1, the neural net was learning only very simple examples, sparse images of white cells. But by grade 6, it was learning very complicated overlapping filamentous structures, and so that helped the overall performance. And we had to do a large search, about 60 days, on a very large system, and it identified one set of parameters, one classifier that we termed Candescence version 1.0. So this is kind of what it would look like applied to a training example. So this is what our labels are, and you can see that basically it gets everything. It disagrees here where it says it's white, not opaque, and we could debate that. Well, one thing, for example, is that it labeled a lot of artifacts, and we tended to get very lazy over time, and we didn't label all the artifacts on the images, but Candescence took that very seriously. And you can see here again that you start to see the SHMU architectures and the overlap, etc., and it does quite well. And a hyphy example, and finally a crazy example that's it actually does pretty well there. I wouldn't want to do that by hand. Well, as I mentioned earlier, overall it gets about a grade of A-, not a B+, not an A. It has some problems, but we do actually argue in the manuscript and give some quantitative evidence that this is a very hard problem, and we think that this is a pretty good first shot at it. Well, for one example here, it seemed to be hallucinating. In other words, it would identify cells and positions of the images that we hadn't identified manually. So we collected all of them, and you can see some of them here. And well, sometimes it was we agreed with Candescence 93% of the time, a posteriorly, and it was only 7% of the time where we thought we were right. So why? Well, sometimes they're very dark images, sometimes it's overexposed, and often it's in very dense images. Conversely, there are places where it misses objects that we labeled. In these images here, Candescence got everything right except what's labeled in red. Sometimes it's about density of the image, that makes sense. Sometimes it's the large filamentous structures, it doesn't get the bounding box regressed properly, and other reasons that are more statistical in nature that we continue to work on. If Candescence does identify the object correctly, it actually classifies it with pretty high accuracy, above 80%. And this is a confusion matrix, so the columns here are the ground truth, that's the reality, and the rows are what Candescence predicted. So if you're on the column, that means it's a good classification. If you're off diagonal, if you're on the diagonal, it's a good classification. If you're off diagonal, then of course this is a mistake. And perhaps not surprisingly, a lot of the confusion exists between white, gray-like, and opaque. And if we go back by hand, which we've done, and we ask, well, could we do better manually? It's pretty hard. There's some examples where we can't really get consensus across the group. So it does seem to be challenging, but it is informative to ask what are the sources of heterogeneity that make this problem so difficult. And certainly technical artifacts, but that's clear. The second thing, though, is that these images that we're taking are snapshots, but they hint at very dynamic processes right underneath of them. For example, we're capturing cells that are going from bud to mature, and we're capturing cells that are transiting from a white to opaque morphology. So yeah, those intermediate cells between the endpoints may not look very much like the canonical morphologies, right? So that's kind of a weakness of our classifier, FCOS classifier-based approach. And so we switch gears, and again, I would recommend going to YouTube and looking at these beautiful objects called Generative Adversarial Networks, or GANs. And we use this to try to capture a little bit more fluidly what Candida morphologies look like. So for example, okay, so what's drawn here, this cartoon, the gray represents a sort of cloud that is what the GAN learns. It's the model the GAN learns of what it means to be an acceptable member of the, a normal morphology, right? So basically these X's here are the real examples that the GAN is trained on, the true images from the Verosina data set. But the gray is what is interpolated, right? That sits between those images. It's basically saying, those images that are sort of within the bounds of normal. And what we can do and that's actually, this gray is what we would call latent space, a visualization of the latent space of the GAN. And we can do really kind of cool things with that latent space. Like we could take two real images, this green and that white, opaque and white cell respectively, and we could linearly interpolate at different points along that path. And you could get something like this, where we have a white cell, and that's a real image, and we have an opaque cell, down here, and that's a real image. But what's going on is basically these intermediates here are those points along that black line. They're interpolations, they're dreams from the GAN, of filling in of what it looks like to transit from the white to the opaque state. And you could do this for other processes. And I have a couple movies I can show you. One is here. This is the GAN dreaming, sort of daydreaming about what it means to be a white cell. So it's going through the sort of that cloud, at least the white part of that cloud, and moving through it. And you can see here that some of those intermediate dreams look a little bit opaque, and some of them look a little bit gray, right? So, and that's that continuity, right? And below here you see the same kind of thing, but for pseudo-hyphae. And now the trajectory there is not some kind of developmental trajectory. It's more a trajectory in this case here that's been pieced together from what it looks like, what it's learned from looking at hundreds of images of pseudo-hyphae. So I'm having something there. So what can you do with that kind of abstract latent space, this gray cloud? And what we're working on now is basically a way of anomaly detection. So I think it's safe to say that it's often the case that you're perturbing some cell genetically perhaps, and when you look at it underneath the microscope, the human eye may not be sufficiently robust to really determine if it's a deviation from normal morphology. And our hope here is that the deep learner with the GAN will be better at distinguishing points like X here that fall outside of that cloud of acceptable morphologies, right? And our first experiments seem to work. Here we have a testing control. So the tests here are, in fact, double knockouts. I won't go into the details of those, but and the blue are just basically wild type Candida cells. And we can start to see that the distance from those cells are actually, the distribution of distances of those cells that are abnormal are larger in distribution than the wild type cells. And so what we're working towards basically is this automated anomaly detection. Okay, so yeah, that's work from my lab and our students with Malcolm Whiteley's lab and Vanessa Demeaux and Chris Law at Concordia. Thank you.